So Aaron, you and I are the only guys here?
Hi, at least on camera. Looks like some others are here.
As you can see, I'm from St. Louis. I'm a kindergarten friend of McFinn, the 42nd reversal of ALS as diagnosed by Rick Bedlack at Duke.
Oh, I attended one of his seminars.
Yeah, he's kind of like a rogue neuroscientist that will admit that people reverse that disease. You know, I've known this guy 66 years, and, you know, as sketchy as our pasts have been, he just presented at Salt Lake City for Healing ALS as one of the 8 or 10 reversals they gathered to speak at a Healing ALS conference over the weekend.
Yeah, I guess the question that these things raise is it a different form of ALS or something else, some other motor neuron disease, or what does it teach us? But it's interesting.
Yeah, he had polio in first grade. I mean, he was the only guy out of 60. I don't know if you know anything about St. Louis, but we still stay friends with our high school friends, our kindergarten friends. It's kind of sick. But
that's great.
Yeah, it's fun. Yeah. Here's your other presenter.
Great. Hi, Seth. Looks like you need to unmute for a second, Seth.
Okay. Am I unmuted now?
Yep, everything's good.
Okay, right. Let me
Thanks
check
for coming.
Share my screen. Right.
Looks good.
Good. Okay.
Great, thanks. Bingxing, hi. You're still muted. On the lower left.
Okay, that's fine. Okay.
Yeah, great. Thanks for coming.
Okay, good to see you.
Can you test your slides?
Yeah, sure.
Looks good.
Okay, yeah, it looks good. Be fine.
And then can you stop sharing whenever you're ready? I just have to send up a introductory.
Okay.
Bingxing, I'm in Zhejiang province now.
What?
I went for a few days.
Okay.
Hi, so long.
Hi, Zero.
Hi, Bingxing. Hi, Seth. Nice to see you.
So long. I went to Morgan for a couple of days.
Morgan?
Morgan in
Morgan,
near Hangzhou.
yeah.
Yeah.
I saw a background. It must be somewhere like a full of bamboo. Or,
Yeah, I rented like a Airbnb cabin in the mountains. Yeah.
cool. It's a long holiday.
Yeah. The kids have school on Thursday. Their school doesn't get off all week. So we have to get
Okay.
back tomorrow. And I like how you just switch working on the weekends to make up for the whole week off. The neat strategy.
Yeah. I started working sometime from today and tomorrow.
Are you back in Shanghai?
Yeah. I'm back in the first.
Okay.
And when the huge amount of tourists fill out Qingdao.
Yeah.
Have you tried slides, Bingxing?
Yeah, they both tried.
Cool. I heard so much about you, about the furious debate over last couple years.
Okay, well,
Yeah.
I'll give the latest.
Sure, great. So, yeah, looking forward. All right. So your kids enjoy Shanghai so far?
Oh yeah, they love it.
Oh, you got a lot of friends here at school?
Yeah, friends. They ride their bike to go to the mall with their friends, or they can buy things on their phone. And in America, we didn't want to get them a phone until they were like 21 or 22. But here you have to get them a phone. So they got a phone, and yeah, it's safe. They can go anywhere they want. Their school is nice. They like their school.
Cool.
Yeah, so they're doing well. They're in America. They did fencing, so they joined a fencing team here. They've been fencing a lot.
Oh. Yeah, it's holiday. Usually a lot of graduate students will coming, but this is a holiday, so I'm not sure how many graduate students will wake up early.
Yeah, you were 4 days, right?
Anyway,
Yeah, they should still attend.
yeah.
Do the grad students really leave Shanghai for the holiday?
Some.
I think they can.
Yeah.
Okay. Seth, I was looking over your CV. Is it true you had 27 papers as a grad student?
Yeah,
Something
that's
like that?
correct.
Wow. Yeah, the grad students should attend. Now the grad students complain about having one paper. At 27, amazing.
Well, yeah, and they all came in the last 2 years. So it was
Wow.
5 years of nothing. So
Wow. That's amazing.
I learned to defer gratification and to
I
change
haven't learned
my
that.
project.
Yeah, now I'm on some committees where it's just like pulling teeth to get one paper.
Mhm.
All right. To get started.
Yeah,
Yeah.
yeah.
Sure, sure. Mhm.
Okay. Welcome everyone. This is Chinese National Day Week Neuro Zoom edition, broadcasting from Zhejiang province. Welcome everyone. We have great talks coming up. Please tune in next week, and we will hear Qingfeng Wu and Jacqueline Bray give talks, and we're still looking for volunteers for you to present your research. So contact us. If you like watching the talks on YouTube instead of Zoom, they're live broadcast on YouTube, so you can join there and subscribe to the channel. And now we'll get started with Bingxing. Zilong, you can introduce
Sure.
him. Thanks.
Sure, sure. So welcome everybody in the holiday season in China. So I'm very happy to introduce the Bingxing Pan, so who is now a professor in Nanchang University. So Bingxing got his PhD from back 2004. It was first military medical university. Now it's called Southern Medical University. So after a PhD, he went to US, did a postdoc in MH. After postdoc in 2009, he come back to Nanchang University, started his own lab. In his lab, he is very interesting in particularly anxiety-related neurocircuitry. So, over the years, he has many interesting discoveries. Today, he will share us latest finding interplay between the BRA to the BNST pathway for elaborate control of anxiety-like behavior. Okay, welcome, Bingxing.
Okay, thanks.
Yeah. The
Okay.
slideshow mode.
Yeah, it's okay, all right.
Yeah.
Okay.
Slideshow.
First, I'd like to thank for Alan and Zilong's invitation and the kind introduction. So today I'm going to talk a short story about the regulation of anxiety-like behavior in mice by BNST and beta nuclear striatal terminalized BNST circuit. So this
Bingxing,
work
you put a slideshow model and not slide. Yeah.
is okay, right?
No, not slideshow yet.
Ready? Let me show.
Yeah. It's supposed to be. I don't know. Aaron, you didn't see the slideshow, right? Not full screen.
Yeah, maybe.
It's not
Okay,
in
there you
full
go.
screen?
Yeah.
It was
There
right?
we go.
Okay,
Yeah. Go ahead.
Okay. So this work was completed by Dr. Ren Wenhai in my lab and 3 students in my lab, Zi Yang and Chun Jiao and Zeyu Hu. So we all know anxiety is a physiological mood when we are in face of uncertain conditions. Say if we have a job interview tomorrow and we don't know we can get a job smoothly, and maybe we will feel anxious. However, when we finally get a job smoothly, and the anxiety will be gone. So unfortunately, more and more people in the current society are suffering from anxiety disorders. And they say they are suffering from excessive and long-lasting anxiety. And this is anxiety disorder. So anxiety is now one of the most prevalent mental disease. And particularly in the last 3 years, and the COVID-19 pandemic time, and more people are suffering from mental diseases like anxiety disorders and depression. So now, understanding how anxiety is formed, regulated, and become dysregulated, and find ways to prevent or treat mental illness such as anxiety disorders is now became big challenges in neuroscience and psychiatry. So research in the past decades have shown that anxiety is regulated by multiple regions in brain, particularly in the limbic regions areas and the complex circuits established among these regions. So in my lab, so we focused our research interest in understanding the amygdala circuit, cellular and molecular mechanisms in the physiological and pathological anxiety. So maybe we all know, and amygdala is a nuclear complex consisting of more than 10 subnucleus and located deep inside the temporal lobe, and that has a critical role in regulating the mood of anxiety and fear. So more specifically, so we focused our research interest in the BOA, the basal lateral amygdala. So it is all known that the basal medial amygdala, the function of this basal medial is mediated by the complex circuit established by this region with other surrounding areas like, say, hippocampus, entorhinal cortex, prefrontal cortex. And today I'm going to share a story. So we just recently finished between the BOA and the BNST. Actually, the two regions are not located in the limbic regions and are well known for so critical for anxiety regulation. And also there's evidence showing that the BOA send a lot of projection to BNST. However, how the two regions are coordinated to fine-tune anxiety is largely unknown. So first, we are going to dissect the projection of BOA neurons in BNST. Cause, you know, a lot of research shows the ABOA, anterior part of BOA and the posterior part of BOA differs in terms of their molecular expression, circuit organization, and function. So we are going to dissect the projection patterns of ABOA and versus BBOA neurons in the BSA. So for this purpose, we just inject, use the AAV strategy, and we just expressing the EYFP expressing AAV into the anterior ABOA and the actually expressing AAV into the PBOA, and see how their fibers goes in the BNST. So we find the terminals, the fibers from the anterior part of BOA, so most likely located in the anterior dorsal part of the BNST. And then most of their fibers goes to the anterior dorsal part, with very, very few seen in the ovum part of BNST. But for the posterior BOA, we can see the fibers goes to both ovum part and the anterior dorsal part of it, with most of them located in the ovum part. So we can see here that the anterior part of ABOA and the PBOA have different projection patterns in the BNST. So this result was further confirmed by the retrograde tracing, and when we inject retrovirus expressing the actually in the ADBNST, we can see the neurons mostly located in both. You can see located in both ABOA and the PBOA. However, when we inject virus in the ovum part of BNST, and we can see the BOA neurons was selectively located in PBOA, but very, very few, only very few seen in the ABOA. This is consistent with the last, the above kind of anterograde tracing experiment. So based on above finding, we can see the ABO and the PBO neuron have different projection patterns in BNST. So we next ask how the ABO and the PBO inputs regulate the activity of the target cells in BNST. So for this purpose, we combine the optogenetics and size coding in BNST. First, we express the triple reduction in the A and the BPBO inputs, and we made a whole cell coding in the BNST, and then we injected a small depolarizing current to evoke the firing of the record cells at a very low frequency, about around 3 Hertz. And then we just deliver the trains of light pulse to the BNST to activate the inputs from the anterior and the posterior BOA, and see how the activation of the A or PBO inputs to affect the firing of the BNT cells. Then the stimulation was stopped. So we find that in the ABL pathways to ADBNST and the pathways from PBLA to OVPNST, we can see activation of the inputs, BL inputs, can both increase firing of the recorded cells in ADBNST and the OVBNST cells. So this is not surprising because we know that the inputs from the BLA, either ABLA or BBLA, are glutamatergic and excitatory. But to our great surprise, in the pathways from PBLA to ADBNST, we find the activation of the PBLA pathways does not excite the cells. Instead, they inhibit, suppress firing of the recorded cells. This is really quite surprising because we know, as I mentioned, the PBL fibers are also excitatory. So how can the PBL activation can inhibit the firing of ADBNST cells? So we think one possibility is that the activation of PBLA can cause a dramatic shift in the excitation and the inhibition balance to the more inhibited state. So for this purpose, we directly cause the response, the synaptic response in the 3 pathways in response to the increased light stimulation. And we find when we stimulate the 3 pathways, we can record the excited postsynaptic currents in the 3 pathways. And the current in the PBLA to OV pathways, the current is very huge. This is also not surprising because I just mentioned there's very lot of fibers in the OV BNST related to the AD pathways. But to our surprise, when we record the iPSCs, and we find that in the ABLA to AD pathways or the PBLA to OV pathways, the iPSC amplitude is really proportional to the EPSC. But in this particular pathways, in the PBLA pathways to AD pathways, the iPSC is really huge. So as a consequence, we can see the IE ratio in this particular pathway is much higher than the other 2 pathways. So we can say, oh yes, there's really the PBLA activation really evoke a very huge iPSC in this AD-BNC regions. So also this is the stronger iPSC really accounts for suppression of AD-BNC neurons activity by the PBLA input. So when we add the PTX, the GABA A receptor antagonist, and this antagonist can really reverse the suppression of the activity of AD-BNC by the PBLA. And we can see in the presence of PTX and the activation of PBLA does no longer inhibit the firing of ADBL. Instead, they just excite the cells. So we next ask where the source of the stronger iPSC in ADBNC cells by PBLA. We think there's 2 potential sources. One is the local GABAergic circuit. That's to say the PBLA activation can recruit a very strong GABAergic circuit in the local ADBNC circuit. And the second possibility is there's activation of PBLA pathway can recruit the GABAergic circuit from other regions, say from the OVBNC pathways, because this part also there's some GABAergic projection to the ADBNCT. So to differentiate the 2 sources, and we just use the particle system to locally, activate the PBLA inputs in the ADBNCT. And then we move the light spot to OVBNCT. And then we can see when we put the light spot in the ADBNCT, so we can see, yes, they can evoke very clear the iPSC. But when we move the light spot to the OVBNCT, we can see much larger iPSCs. So generally we can see, okay, yes, the iPSC in the ADBNCT evoked by PBLA, there's 2 sources. The one source is from the ADBNCT and the other is OVBNCT. And the sources, so the OVBNCT can generate a more stronger iPSC relative to the ADBNCT. So we next check how the inhibitors generated by the 2 regions can affect the firings of ADBNCT by the PBLA. So we can see when we stimulate only selectively in the ADBNCT, we can see that this stimulation can excite the cells firing, and not like the whole activation of the whole PBLA inputs they suppress. this is quite opposite. However, when we just stimulate the fibers in the OVBNST, and we can see yes, in this pathways we move the light spot, and this activation of the input in the OVBNST really just kind of suppress firing of the record cells in ADBNST. So localize the inhibition generated by the OVBNST is really required for the suppression of ADBNST neurons by the PBOA inputs. So just to give a very short summary of our above findings. So we find that the ABOA and the PBOA, they send different, their projection patterns in BOA is quite different. That it says ABOA sends the fibers mostly to the ADBNC parts, and the PBOA sends their fibers to both, to either ADBNC and OVBNST, and with most of the fibers in the OVBNST. And those two subregions of BOA also have different effects on the BNST microcircuit. And the ABOA can kind of, excite the BOA, the ADBNST cells, and the PBOA can just suppress the firing of ADBNST cells. So given this, we next ask what's the specific roles of the A and the PBOA BNT pathways in anxiety regulation. So for this purpose, and we just use kind of opportunity to selectively activate the fibers from the ABOA to ADBNST or the PBOA to BNST. And we find that if we activate the fibers from the ABOA, the fibers from ABOA, and we can see the mouse, when we test the anxiety behavior, particularly in the open field test and in the elevated primase, we can see this kind of stimulation can increase the time mouse spent in the center regions of open field, and increase the time mice spend in the open arm, and increase the time, the entrance of the open arm. So say that the activation of this particular pathways can induce an anxiety effect. However, when we activate this particular circuits from PBOA to DBNST, and we can see they actually induce an over anxiogenic effect. So that is to say, activation of this pathway can decrease the time mice spend in the center region in open field test, and decrease time the mice spend in open arm, and decrease the entry times. So also, because our above findings also show that PBOA send two pathways to BNST. One is to kind of ADBNST, and the other is from OVBNST. So we next ask what's their specific roles in anxiety disorder in this kind of sub-microcircuit. So for this purpose, we try to understand whether or not the neurons from BBOA to the two subregions are the same population or different population. So for this purpose, we just using kind of a natural AV strategy, and we find the neurons innervating the ADBNST are largely from the medial part of the PBOA, and the neurons innervating kind of OVBNST are largely from the lateral part of PBOA. They are quite different kind of space. They kind of have a different spatial distribution in PBOA. And this result was further confirmed by the anterograde tracing experiment. That is say when we expressing the EYFP expressing AV into the kind of lateral part of PBOA, and we kind of fibers almost goes to the OVM part. We can very few in the AVM part. Instead, when we inject the virus into the media part, we can see the fibers goes to mostly to the ADBNST, but very few things in OVM part. So actually, even for the PBOA innervating AD and OVBNST, they are also from have different sources. So same, we just tested using kind of optogenetics to test whether or not how the two pathways participated in regulation of anxiety behavior. And then say when we activate the pathway from media part to the OVM kind of ADBNST, we can see that is this kind of activation can decrease the anxiety behavior in mice. That is, they can increase mass spending in the center region in the open field and increase time mice spending open in the elevated maze and increase the mice's entry to the open arm. So by contrast, when we activate the LP, the lateral part of BOA to OV BST, we can see activating these pathways can increase the anxiety behavior in mice and decrease the mice spend in the center regions of kind of the open field, and they also decrease the mice spend in the elevated maze in the open arm and the entry times to the open arm in the elevated maze. So we just revise a little bit to our above findings. That says it is where to go determines the specific function of the BOA neurons in anxiety regulation. So regardless of the neurons in ABOA or PBA, when the neurons projection to ADBSD, the neurons are kind of anxiolytic, and however, when the neurons innervate the ovarian part, and they most likely anxiogenic. So finally, we'll ask how the two circuits respond to the changing environment. Say, when we switch, from the anxiolytic environment to the anxiogenic environment, how the two pathways, so the activity of the two pathways were altered. And we can see, so to answer this question, so we just, test the activity of the two pathways in the kind of elevated maze. So we just express the GCAM in the ABOA neurons or PBO neurons, and we check the activity of the fibers when in the anxiety behavior test. So we can see when the mice move from the closed arms to the open arms, we can see the ABOA pathways to ADBSD, this particular pathways, the activity will goes down. However, when the mice move from the open arm to closed arms, the activity goes up. That is when the mouse in the anxiogenic environment, this kind of anxiety pathways will go down. So when we test the other pathways, that is from the lateral posterior POA to OVBST, and then we find that when the mouse move from the closed arm to the open arms, and we can see this pathway goes up. And when the mouse moves from the open arm to closed arm, and the activity of this pathway will go down. This is quite opposite to the ABOA pathways. So based on the above findings, we just propose a working model. Say when the mice in the anxiety environment, the pathways from either ABOA or MPBOA to the ADBSD neurons will be activated and then cause the anxiety effect. And so by contrast, the circuit from the lateral part of BOA to the ovarian PNST pathways goes down, and then will cause the disinhibition of this pathway and finally contribute to the anxiety effect. However, when the mice in the anxiogenic effect and the pathways from APOA and the MPBA goes down, and the pathway from LPBOA to the OV pathway will become dominated, and then will inhibit the activity of the AD neurons, then result kind of lead to the anxiogenic effect. And we propose that the dynamic switch between these two models is really important for the appropriate expression of anxiety under the changing environment. So I'd like to thank the members in my lab and the suggestion and comments from the collaborators from Xiaodong Wang and Tifei Yuan, Haitao Wu, and Andrew Hopkins from NIH. And the funding sources from the Ministry of Science and Technology of People's Republic of China and the National Natural Science and Science Foundation of China and the Nanchang University. So thanks for your attention.
Great, Bingxing. It's very amazing discovery. So for question time. Yeah, I have a quick question for the how do you think this from anxiety to the and kind of anti-anxiety, how the state switch within the BRA, right? So it has to be some balance within the BRA from media lateral part.
Yeah.
So Is there microcircuitry from MP to LP? There's some inhibitory or sanctuary?
Actually, so far there's no direct evidence showing, particularly in psychiatric conditions. So I think our finding is the first piece of evidence showing particularly the two paths will get switched, just in response to the change of the environment. And
right.
So yes, we propose, we think that as particularly for the different subnuclei of BST, and they will respond different to the change of the environment. Say, if the mouse was individual in the anxiety environment, and the activity of the AD part of anterior dorsal part will become dominant, and the OV part will goes down. And so if the individual goes to the anxiety neurogenic environment, so the OV part will goes up, and the other part will goes down. Yes.
Okay. Maybe you can combine your finding with some single cell sequencing data to see some particular subpopulation in the media lateral PBRA, which might be can say molecular markers, right? Is there lot of
there's some, I'm not pretty sure there's kind of, there's a specific molecular marker for the two part, but we know some molecular expression is quite differ between the two regions, very different. Okay. I'm not sure they can work as a marker for the two neuron projections. Is the BNST? I don't know. Yes.
Sure. Yeah. David, you have a question?
Yeah. Little slightly off topic, but do you know if during development that initially these projections get intermixed and then refine into their separate anxiety versus not over time, or is it kind of hardwired?
Actually, I have no idea how the kind of circuit is get established during development, and we just do some kind of check some changes during the development of some kind of anxiety disorders. And we just find that there's different changes in the two pathways. They really got different changes. But for the development issue stuff, I have no idea. Sorry.
How early do you think it's feasible to trace those projections?
How early? I think it may be at least as kind of, because this pathway became mature at the kind of adolescence, I think. Yes.
Thank you.
May I ask a question?
Sure, go ahead.
Yeah, Professor Pan, that's a great talk. So I'm wondering, when you do the retrograde tracing in the bad nuclear, did you see any back label cell in the central amygdala?
Oh, I have to double check whether or not there's some neurons in the central nuclear amygdala. Actually, we know there is some projections from the BST to central amygdala. I know, but actually, I really missed this point. Sorry for that.
Okay, no worries. Thank you.
Yeah, I have to double check whether there's some neurons in the central amygdala. This is quite important. Okay. Very interesting. Yeah.
Yeah. Thank you.
Okay, is there any more questions? Okay, I have last question about how the animal decide to be access or not. Is there a switch within the PBRA, do you think, or related to the upstream of the ABRA, PBRA?
Actually, definitely, I think the signal mostly come from the upstream, because we have traced the upstreams. The two subregions receive different inputs from different regions. Yes.
Great. All right. So some more questions. Thanks, Bingxing, for amazing talk.
Thank you.
Aaron, so you should go ahead to introduce Seth, the next speaker.
Great. Thanks so much. So it's a pleasure to introduce our next speaker, Dr. Seth Blackshaw, who is a professor at the Department of Neuroscience at Johns Hopkins University. Dr. Blackshaw did his undergraduate degree at University of Chicago, and then moved to Johns Hopkins, where he did a PhD with Dr. Solomon Snyder. And as a graduate student, Seth had 27 papers, which I think is unprecedented, especially in this day and age when it's hard for grad students to finish with one paper. He then moved to Harvard Medical School, where he did a postdoctoral fellowship with Dr. Connie Sepko. And here in a paper published in Cell, he used this ancient technology from the 90s called SAGE, which is serial analysis of gene expression. It's actually an amazingly powerful technique in which you use sequencing of cDNA and counting to define gene expression. And here he did this in photoreceptors and discovered new genes expressed in photoreceptors, some of which are human disease genes. He then returned to Johns Hopkins University, where he started his own laboratory, and he has made major contributions to defining mechanisms that control generation of different cell types in both the retina as well as the hypothalamus. And we're delighted that he came here today to share his latest results with us. Thank you, Seth.
Well, thank you very much, Aaron, for the invitation. I see a bunch of old friends in the audience, so thanks to all of you for coming as well. And can you see my screen?
Not yet.
Okay, let me share. Can you all hear me?
Yes.
Okay, great. Thank you. Okay. So what I'm going to tell you about today is our group's work on the regulation of neurogenic competence in retinal glia. And the retina is, as Aaron mentioned, I've been studying ever since I was a postdoc. It is the most tractable and accessible part of the central nervous system. It's a fantastic model for studying the brain, and it's extremely evolutionarily conserved, as you can see from human and mouse H&E stains here. Now, much of my talk is going to focus on retinal glia, which it's a rather unusual form of radial glia. They basically do the work of gray matter astrocytes in the retina, but they are morphologically and in their gene expression profile very similar to retinal progenitors, with the big difference in mammals being that they don't divide and they don't make any neurons. But they're of considerable interest from the standpoint of potential cell therapy. So the selective death of rod photoreceptors, which sense dim light, and cone photoreceptors that sense bright light, is the reason behind loss of vision in hereditary diseases such as retinitis pigmentosa and photoreceptor and macular degeneration. These are selective neurodegenerative disorders in just the same way as, say, Parkinson's is, except they're confined to the retina. Loss of ganglion cells, which convey information from the retina to the brain proper, also mediate or underlie glaucoma. So there's been a lot of interest in potential means of taking these progenitor-like cells and using them to regenerate one or more of these other cells that are lost in disease. I want to give a brief mention to two types of retinal interneurons, bipolar cells that relay information from photoreceptors to ganglion cells, and amacrine cells that modulate the bipolar to ganglion cell synapse, as they will come up later. Now, back to glia. Now, they're not progenitors in mammals, but they actually are in most vertebrate species. Most cold-blooded species, including zebrafish, any form of retinal injury will stimulate glia to rapidly dedifferentiate, acquire neurogenic competence, re-enter the cell cycle, proliferate, and generate essentially every form of retinal neuron. And over the course of days to weeks, completely regenerate all cells lost due to injury in the retina. Now, this remarkable ability has been lost repeatedly over the course of evolution, and it's been lost separately by both birds and mammals, although juvenile birds retain a very limited level of neurogenic competence. Glia of mammals, however, unfortunately lack any ability to generate neurons whatsoever, and instead undergo reactive gliosis following injury, followed by a gradual return to quiescence. Now, there's really three main mechanisms to keep in mind when developing strategies for reprogramming glia for therapeutic purposes. One approach, and this has really been the most popular, would be to overexpress neurogenic factors, so neurogenic BHLH factors, for instance, in glia and turn them into something more like a neurogenic progenitor. Another approach might be to go turn this on its head and inhibit the activity of inhibitory factors that block neurogenic competence in mammalian glia. And of course, once you've made neurogenic progenitors out of glia, you got to guide them to make the types of cells that you want. So photoreceptors, if you're trying to fix a retinal dystrophy, or ganglion cells if you're trying to fix glaucoma. Now, today I'm going to talk about our work on identifying inhibitory regulators of this process, but we've worked in all three of these areas. Now, I want to tell you first about our work on identification of negative regulators of neurogenic competence in mammalian glia, and then secondly, talk about some basic pitfalls we've encountered when trying to apply these findings to induce neurogenic glial reprogramming in vivo, and how we might be able to work around this. So as part of a larger collaborative project sponsored by the Audacious Goals Initiative at the National Eye Institute, we've been carrying out a comprehensive analysis of both gene expression and chromatin conformation in 3 species with very different regenerative competence. Zebrafish, which regenerate like crazy, chick that have a very post-hatch chick that have a very limited ability to generate neurons, and mice that have no ability whatsoever. We have injured the retina several different ways. We've used excitotoxic NMDA treatment to kill intraretinal neurons, light damage to kill outer retinal neurons, growth factor treatment to induce neurogenesis in zebrafish in the absence of injury, and then analyzed changes in gene expression and chromatin conformation in glia at various different time points following injury. We've done this using single-cell RNA-seq, as well as bulk RNA and ATAC-seq from FACS-sorted glia. And then by integrating these data sets, we've been able to identify changes in gene expression that correlate either positively or negatively with regenerative competence. We've then been able to integrate these data to identify regulators, gene regulatory networks that control quiescence, proliferation, and neurogenic competence in glia, and in an unbiased manner, identify genes that regulate these processes. Now, this is single-cell RNA-seq data from whole retina following NMDA treatment in mice, zebrafish, and in chick. And here I've highlighted the glia. We can readily distinguish resting glia in orange and then activated glia in red. Now, all 3 species show this transition from rest to activation following injury, but how they behave after that is very different. So in the mouse, here we have the glia at rest, and here we have the glia 3 hours following injury. The biggest shift in gene expression occurs in the first few hours after injury. And then the glia gradually return to a resting state. So none of them proceed to become neurogenic. That's very different in the fish. In zebrafish, again, you have a transition from arrest to activation, but then most of the glia, after about 10 hours or so, transition towards a proliferative and neurogenic progenitor-like state. Chick are somewhere in the middle. Essentially all of the glia reactive by about 24 hours following injury in the chick. Most of them then return on to resting state, but then some go on to become reprogrammed to be proliferative and eventually neurogenic. So by integrating RNA and ATAC-seq data, we can identify gene regulatory networks in glia, in zebrafish, and mice, and can identify some clear similarities and differences. What I've done here is taken a very complex hairball diagram that summarizes transcriptional regulatory relationships between individual transcription factors that are active here in the rest state, in an activated and then an activated or gliotic state. Transcription factors in the rest network activate other transcription factors in the rest network and maintain this state, and also directly inhibit expression of transcription factors in this activated or gliotic network, and vice versa. So you got this biphasic, auto-activating, cross-inhibiting set of transcription factors controlling the transition from rest to activation. So this is a common theme that you see in developmental transitions. And this is the case for both mouse and zebrafish, but then things get very different. So following activation, most of the cells then quickly reprogram to a proliferative neurogenic state. In the mouse, on the other hand, we have that activation of what we've termed a restore rest network. This largely consists of transcription factors that are active during rest, whose expression goes way down following activation, but then after about 10 hours comes shooting way back above baseline. And these return glia to a resting state. So these are the differences. So this actually makes a couple of predictions. The first prediction is that in order to reprogram, glia will have to pass through an activated state. We've tested that in zebrafish by disrupting the activity of transcription factors in this network, and that blocks proliferation and reprogramming. The question arises, why doesn't this happen in mice? Well, there's a couple possibilities. One is that this activation or gliosis promoting network is fundamentally different. It turns out that it is. That in particular, notable difference is that the NF-kappa B pathway is far more active in an injured mouse glia. So this shows you genes from the NF-kappa B pathway activated following different time points following an NMDA injury. This is the mouse. You can see a lot of genes are activated and stay on. This, in contrast, is the zebrafish. Very few of the NF-kappa B pathway factors are activated. Most of the ones that are activated are actually negative regulators of the pathway. You can see that the chick is really kind of in between, a little more, partially mouse-like, partially chick-like, or fish-like. So working together with Andy Fisher at Ohio State, we tested whether the NF-kappa B pathway might be actively inhibiting proliferative and neurogenic competence in the chick. To do this, we treated with sulfasalazine, which is a broad spectrum inhibitor of NF-kappa B signaling, and induced neurogenic competence by treating with FGF2. And we found that sulfasalazine treatment leads to a dramatic increase in glial proliferation and eventually neurogenesis. In contrast, it inhibits proliferation of microglia, and injury induces the NF-kappa B pathway just as efficiently in all three species. So a second potential means for inhibiting neurogenic competence is the induction of this restore rest network. And here, to study this, we focused on three members of the NFI factor family, NFI A, B, and X, which are highly expressed at rest and then also strongly activated in this restore rest network. So to study the effects of selectively disrupting the NFI factors in mature muloglia and to simultaneously analyze the fate of NFI deficient glia, we used tamoxifen-inducible Cre recombinase to both activate expression of a SUN1 GFP reporter that outlines the nuclei of muloglia, and then also monitored the fate of GFP positive cells in both wild control and NFI deficient retina. So here we've injured the retina, we've introduced essentially inflammation and reactivity with NMDA and growth factor treatment. In the controls, we see no glial cells whatsoever. In the NFI triple mutant, we see about 15% of the GFP positive cells are labeled with CRX, selective marker of bipolar and photoreceptor cells, and 15% labeled with HUCD and NUN, which in the retina are markers of amacrine and ganglion cells. So we are clearly inducing transformation of glia into neurons by loss of function of the NF5 factors. And we can visualize this using single cell RNA-seq. These are GFP positive cells 21 days after NMDA treatment in control, and NFI deficient cells. Most of these cells are glia, but we actually see a neurogenic differentiation trajectory, giving rise to bipolar-like and amacrine-like cells. We can visualize these bipolar and amacrine-like cells by infecting with a Cre-dependent M-cherry AAV. Most of the, about 70% of the cells labeled in the NFI mutants are muliglia-like, 15% are bipolar cell-like, 15 amacrine cell-like, and we can see that these express protein markers of bipolar and amacrine cells. So to summarize this part of the work, this part, which is all published, we found that we found in many ways a parallel organization of the gene regulatory networks in fish and mice following injury, but that neurogenic competence is actively inhibited in the mouse by premature activation of this restore-rest network, and also by activation of by excessive activation of inflammatory signaling through the NF-kappa B pathway. Loss of function of the NFIs gives rise, induces glia to generate interretinal neurons, including bipolar and amacrine cells. So a lot of unpublished data that I don't have time to show you. It's big issue here is that we're not making either photoreceptors or ganglion cells. We're working on that, and I can talk about that in the Q&A. But this basic research is all well and good, but what we'd really like to use our findings to do are to reprogram glial cells in the human eye to replace neurons lost due to neurodegenerative disorders. For this, we can't rely on transgenesis or conditional mutants, as we've done in the studies I've described so far, but instead we'll need to use viral vectors and small molecule treatments. So in the last few minutes, I'd like to shift gears and discuss some serious technical pitfalls that face some of these studies, how they might arise, and one way we might address them. So I think many, if not most folks know that of two recent studies that claimed to observe a full conversion of retinal glia into retinal ganglion cells and brain astrocytes into dopaminergic neurons, midbrain neurons, following knockdown of one factor, of one gene, the splicing regulator PTBP1, which is expressed in progenitor cells and bipolar and astrocytes. And both of these effects were reported to occur independent of any injury. So this looks basically like a miracle. There was a lot of fanfare, publication in high-profile journals, but there were several major red flags raised by these studies. So first, both studies used infection with AAV GFAP mini promoter constructs, expressing Cre recombinase to infer cell lineage, rather than doing prospective cell lineage analysis using tamoxifen-inducible Cre line, transgenic Cre lines, like I showed you previously. So which is really the gold standard in doing cell lineage analysis in developmental neuroscience. Secondly, no clear evidence was provided for the existence of immature or transitional states using either molecular markers or high-resolution morphological analysis. And finally, both studies involved knockdown of PTBP1 rather than a bona fide genetic loss of function, raising questions about specificity. In fact, last year, Chun-Li Zhang's group at UT Southwestern reported that astrocytes labeled using a tamoxifen-inducible Cre line showed no evidence of conversion to neurons following PTBP1 knockdown, or for that matter, NeuroD1 overexpression. So we took this work one step further by combining prospective cell lineage analysis with glial-specific deletion of PTBP1. Using the same GLAST CRE-ER transgenic line and Cre-dependent SUN1 GFP reporter I described earlier, we conducted glial-specific loss of function of PTBP1 in adult retina. And as you can see, we were able to very efficiently eliminate PTBP1 expression as measured by immunostaining, and at the same time, label glia. However, none of the glia turned into neurons, none of them expressed neuronal markers, none of them lost glial markers. And this occurred both in an uninjured state and following destruction of the great majority of the retinal ganglion cell layer by excitotoxicity. We saw the same thing. We essentially saw the same result in the brain when we deleted PTBP1 in astrocytes with the tamoxifen-inducible Cre line, the ALDH1L1 Cre-R line, and at the same time labeled these cells. So again, we were able to efficiently eliminate PTBP1 expression, but we never saw GFP-positive cells expressing neuronal markers, and they did not lose glial markers. Single-cell RNA-seq analysis of purified cells likewise shows no changes consistent with a cell phase switch or a loss of glial identity. So what's going on here? So what might explain the data obtained from the AAV GFAP mini promoter constructs, which seem to show direct and robust conversion of neurons into glia? So we got some insight into this problem by performing some of our own experiments using this same construct. And here, what we were trying to do was misexpress transcription factors that we predicted might convert glia to neurons. In here, we used a P2A line to express M-cherry and either one or two different transcription factors. So, but at the same time, we conducted we labeled these cells with the class Cre-R M-cherry, or class Cre-R Sun1 GFP before we infected with a virus. And what we saw was really unsettling. When we infected with just an M-cherry GFAP reporter alone, we labeled only glia. This was also the case for two of our transcription factor constructs, OCT4 and NRL, just labeled glia. But then for 5 of these constructs, not only did we lose glial expression of M-cherry, but we now activated expression of M-cherry in intraretinal neurons. Now, if we hadn't pre-labeled these cells with GFP, we would have concluded that we had converted glia directly to neurons. But none of the GFP positive cells ended up labeled with neuronal markers. So what this tells you, the AAV serotype used here infects both neurons and glia. And what this tells you is that the GFAP mini promoter is sensitive to the insert. Is insert sensitive. So different inserts produce very different patterns of reporter expression. And this tells you basically that the GFAP mini promoter is useless for any studies used to infer cell lineage, and should not be used in this context at all. I think this also raises some important questions about mini promoters in general. If their specificity can vary dramatically given the context, given the insert that's present, what can we do to control them? What can we do to make them more specific? So we can't combine different cis-regulatory elements in any predictable or reproducible way, but we can use alternative and complementary approaches to improve specificity. So our lab has made use of highly cell type specific patterns of alternative splicing to develop what we've termed splicing linked expression design, or SLED-based AAV vectors. These constructs use cell type specific patterns of alternative splicing to induce translational frame shifting, thereby restricting expression of a construct of interest to a specific cell type in which both the mini promoter used to drive expression and the alternative splicing pattern are active, effectively acting as a Boolean AND function. So I want to show you just some examples of how this works. This is the ubiquitously expressed PLS3 gene. It has a neuron-specific mini axon that is evolutionarily conserved. It's found in both mouse and human. So here we've taken this mini axon and we've hooked it to a ubiquitous promoter, and we've used it to drive a bichromatic reporter such that GFP If this infects a neuron, it will be infected with GFP. If it infects any other cell type, it will express DS-RED. And this shows you how this sled vector acts or works. So it gives you very specific neuronal GFP expression, but it expresses the alternative reading frame in glia. Now, we can do the same in the retina for photoreceptor and non-photoreceptor cells. And this shows you an example of how this is used in a Boolean fashion. So here we have, this is the ENS reporter. It uses an intron in an alternative exon in the SynRG gene that's specific to excitatory neurons and glia. We hook this to the ubiquitous or to the pan-neuronal H-syn mini promoter, and voila, we have an excitatory neuron-specific AAV reporter line. So this work just came out in Nature Communications. We're now refining these vectors to generate improved vectors for glial-specific expression. So I'd like to close, thank the folks who did the work, and the folks in the lab who always make it a fun place to be and our sources of support. And in particular, I'd like to thank Tan Huang and Jay Wang, who did all of the work on Mula-glia reprogramming. Tan will be starting his lab at University of Michigan early next year, and Jay is at Chinese Academy of Sciences in Guangzhou. Niet Le and Haley Apple, two grad students in the lab, spearheaded the work on the PTBP1 knockout and the GFAP mini promoter, and John Ling did worked out the sled vectors. Thanks to our other collaborators, and thank you for your attention, and I'd be happy to take questions.
Thanks very much, Seth. Awesome talk, and we're open for discussion. Dr. Fu.
Yeah. Hi, Seth. Great to see you again. Let me just raise two questions. Have you applied your latest double lineage tracing strategy to deal with the PTB lockdown?
With the PTBP1 knockdown,
yeah.
We have not used it. We have not used the PTBP1 knockdown, though other groups have conducted similar studies.
Okay. So in this regard, have you ever seen any evidence to show the PTB lockdown induced neuron actually is lineage traced to the endogenous neuron? Is there such evidence in the literature?
We have no evidence that PTBP1 deficient neuron, PTBP1 deficient cells generate a neuron. Neuron.
No. I'm asking.
Both for hex and homos.
I'm asking you to, according to your knowledge, whether or not anybody has demonstrated those neurons are apparent converted neuron are due to the endogenous neuron due to the leakage. Have you ever encountered such evidence? If not, then the alternative question is whether a lot of things may not come from Mula-glia or other cells, but come from somewhere. And what's the evidence? I mean, now we just don't know where they come from. Is that right?
I think there is no evidence whatsoever that neurons are generated from following AAV infection of PTBP1 neurons.
Right. So neurons are not generated from the cell you trace. Does not generate from other cell you don't know.
Well,
you cannot trace one cell type to neuron. That means everything is due to the leakage. This has bin demonstrated with Neuro D1, but a lot with PTB.
That's
Number two, let me make a comment here on your genetic lockout, which is no leakage, very robust. According to your own data, PTB lockdown induced NPTB or PTBP2. Do you agree or not?
There is a modest increase in, so I want to follow up on the single cell seq analysis. So we now have single cell seq analysis from purified glia from all three brain regions, as well as from we previously shown from Mula-glia. So there is a modest increase in PTBP1 or PTBP2 in mular glia. We do not see any, we also see a modest increase in cortical glia, but not striatal or midbrain glia.
Okay.
It's a very modest increase.
Okay, let me make a comment here. PTBP1 and 2, at least one of them, you cannot get rid of both because otherwise the cell will die. This is what we know. Everybody have showed that. In the other word, when you get rid of PTB genetically, you have to induce PTBP2, otherwise cell will die, unless you have some magic hand, okay? And now induced the PTBP2 has to be downregulated in order to give rise to neuron, otherwise there's no neuron. In the other word, in this case, knockout is different than knockdown. You prevent dynamic regulation. Just go back, check your data. Nevertheless, let me make a final point. In your published cell report paper, count your own rod cell population. And then we reanalyze your data, your molar glia reduced, and then your rod cell increased. We don't know what's the relationship, but read your own data, okay? That's your data. And your data, plot those numbers in a funny way because your wild type cell, at least 30% of them contaminated with your crystal cell, which you are eliminated. As a result, the cell population calculation is unreliable from your published data.
I'm sorry, I actually have no idea what you're talking about.
Okay, fine. I just want to point
If
that
you'd like
out.
to follow up, please.
Yeah,
please do.
check the number, check the percentage of rod cells in response to PTB lockdown, okay? Just check your own data, you will see that.
If you're talking about the single cell seq analysis,
Correct, that's
you
what I'm
should
talking about.
not use single cell seq to infer cell numbers.
But you
And
did
infer properties
though.
of individual cells.
Okay,
And you can
but
infer, say, the presence or absence of a cell type derived from, say, a PTBP1 deficient
Right,
cell.
okay,
But if one sample has more rods than others, that means that if it has 30% more rods, that means nothing. That could easily be an experimental, that's just a dissociation artifact. You
okay.
see a lot of variation in rod numbers.
Okay, I just want to point it out, and hopefully through the healthy science debate, truth will eventually come out.
I think it will.
Okay, cool.
Hello,
Well, nothing
Seth.
personal. I love to talk to you in person or debate. Last time we did not have good enough time to do so.
No, we didn't. So, yes, please. If you, if there's anything you would like to send, please. If there's
Sure.
anything you'd like to carry on offline, I'm happy to do so.
Okay, great. Yeah, let's maintain that the productive, interactive relationship. That'll be great for the sake of science.
Thanks, Shangzhuang. GC Huawei Mate 20.
Hi, this is Gong.
Cool. Oh,
Yes. Hi, Seth. Nice to see you. Meet you here. Yeah, I mean, I'm not going to get into any discussion on the PTBP1. Shangdong has already said enough. I'm interested, like you also mentioned, Neuro D1, as I have already found out that Chinese paper need to reduce that titer. But my question to you is, have you ever done single cell analysis when you overexpress Neuro D1 in the mular glia? Did you see a clear change that the transcription change toward the neurons?
We've done that. So we miss, as I showed, so when we misexpress just with the GFAP promoter alone, GFAP NeuroD1, we see that the reporter shuts down in glia by 8 days following infection and now comes up in ganglion and amacrine cells. So it's interretinal neurons. So none of those cells are lineage marked. We have gone back and we have, and if we use a flexed virus instead. So we've now gone and we've repeated all the studies I just showed you with flexed viruses. where we can selectively, we can restrict expression just to glia. So some of those actually look promising, but NeuroD1 does not. NeuroD1, we don't see any conversion of glia to neurons. That said, we have sequenced those. We've purified those glia out and sequenced them. And there's some rather modest induction of some other neurogenic factors, such as ASCL1, but it's pretty minor. So I would say, yes, it has the capacity, it has the ability to induce a small subset of neurogenic genes, but those cells do not in any obvious way differentiate into neurons.
So you might have seen our bioarchive paper. I suggest you put an enhancer in your NeuroD1, and some of your the IPRM cherry labeled the lineage trace astrocyte will be converted into neurons. If you only use a weak GFAP promoter, you may not be able to convert those lineage trace astrocytes. But those non-lineage trace astrocytes or Muller glia in a retinal case, you will see it will be converted. We will have some evidence showing you that Muller glia, such big cell soma, when you add NeuroD1 or other transcription factor, you will see them actually move up and down. It's very fascinating. I'm sure you are the expert on the retina. I'm not, but when I see that Muller glia cell body move up and down, I mean, that's a clear evidence they are converting. Another question might suggest to you is, do the RNA-seq between pure wild-type astrocyte or maybe wild-type Muller glia in your case, and those lineage trace, the Muller glia, you will find they are different. So as I point out in my paper, that those lineage trace, the astrocyte, or perhaps in your case, the Muller glia, their whole transcriptome is different from the normal wild-type one. So I guess when you have those, tamoxifen-induced Kryllox-P recombination, it actually changed a lot of epigenetics of those lineage trace astrocyte or Muller glia in your case that can make the conversion a little bit more difficult.
Well, we have seen that. We have seen that with some other factors, which I can't name at this point because that's still, though that work is still unpublished. We have not seen it with either ASCL1 by itself or NeuroD1. So again, a little bit of induction. Now, what we have found that, yes, drugs, small molecules that modulate that basically increase chromatin accessibility help facilitate induction of neurogenic genes. We've not tested that for NeuroD1. That may be a possibility. That said, I mean, I think the thing that worries me much more about NeuroD1 is the fact that the GFAP mini promoter not only shuts down in glia, but actually comes on in neurons. And this is the work that's been done previously. This is how lineage analysis has been done. So you need to trace the cells before you infect.
Yes, as we have already shown, even in lineage trace astrocyte, when you use enhancer to increase NeuroD1 expression, you can convert those lineage trace astrocyte into neurons, right? We have already shown that. So perhaps I'm just suggesting you in retina also use the enhancer, to NeuroD1 AKI-SCU1 and see, they might show difference than you expected.
Yeah, it depends. I mean, we've used the we've done the NeuroD1 with the EF1 alpha flex. That's a fairly strong promoter, but yeah, we could make it stronger.
EF1 alpha actually is weak. In our hand, EF1 alpha comparing to CMV enhancer, at least in the cortical astrocyte, EF1 alpha is much weaker. The CMV enhancer is much stronger.
Yeah, it's at least in Muller glia, it's EF1 alpha over CMV over CBH. Is basically
simply a suggestion that if you use CMV enhancer, Yeah, I'm just give a suggestion. CMV enhancer, you might see difference. And the GFAB promoter, those GFP, Neuro D1 GFP, surely they will be downregulated actually. You will see what we have seen already. We also are doing some retinal and neuronal conversion as well. It is indeed those GFAP, Neuro D1 GFP, in certain time, I believe somewhere 20 days, 30 days, they are actually downregulated. So there are certain time during the cell conversion, there are a lot of genes will be downregulated, including our Neuro D1 GFP. Then when it turns on in other type of cells, I guess one possibility, as you said, it is leakage, but another possibility, it is genuine conversion. I wouldn't just brush that off, because in fact, when we use a different transcription factor in the retina, we actually see the conversion into different cell types. And we do see the big glia, the Mueller gliosoma is moving up and down. That we will show you after we publish it.
Well, if any of these are actually producing conversion, you would have to assume that the CAG ROSA based reporter we use shuts off in those cells.
Yeah, we will be very happy to try your system as well. We'll be very happy to try that.
Look, if you can prospectively label these cells, you get robust labeling of glia, and then you show that the label ends up in a neuron, and you can show using single cell seq a clear differentiation trajectory, I'll believe you. But otherwise, okay, I'm going to assume it's your wonky promoters. So show me that, and I'll believe you. That goes for everyone listening.
Sure, yeah. Don't dismiss that conversion yet.
Yeah, because that is regenerative neuroscience is conceptually a subset of developmental neuroscience. Those are the gold standards for inferring cell lineage and development. They need to be applied here.
So agree, but let me just make the one last comment that the lineage traced glial cells are for the purpose of lineage tracing. They are not for the purpose of conversion. When we act on conversion, we first need to understand those lineage traced glial cells. What happens to them? What's the difference between those lineage traced glial cells versus normal non-lineage glial cells? That's my advice to you and to the whole field.
Well, we're able to label about 95% of the glia using this reporter. So if you're assuming that the unlabeled glia are behaving differently,
yes,
I can't answer that except to say that they're a very small fraction of glia, and we have absolutely no evidence that they proliferate or behave differently.
Let's try to understand them, Seth. Let's fully understand those lineage traced glial cells and their difference from the normal wild type glial cells. Let's understand them before we make any assumption what is the golden standard, okay?
I think I'm applying the standards used to study cell conversion, to study cell lineage in other systems. So again, I think the gold standards used in the field have to be applied first here. If you can provide any compelling evidence that these unlabeled cells behave differently, I'm all ears. But
okay. So thanks, Gong, for the comments, and Shangdong. Seth, thanks for the discussion, sharing the data, and joining us. And I think it's a good time to wrap up, but I really appreciated the discussion.
Thank you. Thanks very much for the invitation.